My topic is synthesis of immunoglobulins. Immunoglobulins consisting of two types of chains, heavy chains and light chain. Heavy chain consisting of gamma, alpha, mu, epsilon and delta. Light chain consisting of kappa and lambda. Both are linked by disulfide bond. Three set of immunoglobulin chains are present: kappa chain, lambda chain and heavy chains. Chains encoding the variable region of each immunoglobulin chains are present as multiple chain segments. When this kappa chain presents on chromosomal 2 and lambda chain presents on chromosome 22 and heavy chain locus presents on chromosome 14 of an immunoglobulin. Then variable region is present at the end of the each locus of cluster of B gene sequence is present. Its codes for the immunoglobulin chain at presence on B region is present different for different genes. Then leader sequence is present at the 5' end. Leader sequence encodes a signal or leader peptide that facilitates the transport of Ig protein through the endoplasmic reticulum. So before the variable region, leader sequence is present. This diagram shows the pink color shows the leader sequence and variable region is present for in kappa, lambda and heavy chains. Then diversity region is present and joining segments are present. At the 3' end of the cluster of V gene segments, J joining gene segments is present, and it's this is when V and G different for different locus. So the yellow line shows the joining segments of the each chain, kappa, lambda and heavy chains. Then diversity region presents only on the heavy chain. It present between the V and J gene segments of the heavy chains, and it is absent in the light gene segments. So this blue color line represents the diversity region. Generally, diversity regions is of 23 diversity segments are present. Then we move on to the arrangement. Variable region of the immunoglobulin chains encoded by V and J segments in light chain and V, D, J segments in heavy chain, which means diversity region is absent on heavy chain. Then constant region is present. Constant region gene organization is located at the 3' end of the J gene segments of each immunoglobulin locus. In constant region of the kappa chains has only one gene concentration, and lambda chains has a 4 to 5 concentration, and the heavy chains has a 9 concentrations is present, and the 9 concentrations have a sub exons are also present. That is C mu, C delta, C gamma 3, C gamma 1, C alpha 1, C gamma 2, C gamma 4, C epsilon and C alpha 2. These are the 9 concentrations, and the 4 main exons and 2 sub exons are also present, which codes for the heavy chains. Then BDJ recombination occurs by process by which B cells randomly assembles the immunoglobulin gene segments to form the functional variable regions exons. This is called BDJ recombination. So in the light chain, leader sequence, variable region and joining sequence and a concentration is present. First step is somatic DNA recombination, which means gene rearrangement. This was first discovered by the Susumu Tonigawa, a Japanese scientist. They discovered antibody genes can now rearrange themselves within a differentiated cell. Then first step is VJ, V segment, random V variable regions gene segment joined together on the joining segment of the chain. Then second segment is transcription, which means DNA is copied to the pre-mRNA. The formation of primary transcript RNA is formed. The third segment is RNA splicing. Splicing means removal of the introns. Introns are non-coding sequence, which means will be removed, and VJ constant region gets joined. And poly A tail, which means mRNA is formed. Now translations occurs. mRNA is decoded into the light chain polypeptide. So in variable region, variable and joining sequence constant is present. In heavy chain locus வந்துட்டு 9 concentration is present, 4 main exons and 2 sub exons is present, 2 rearrangements even, DJ joining and VDJ joining is present, and VDJ is present only on the heavy chains. So in first step of first somatic DNA recombination occurs. Randomly variable regions joined together to the diversity region of a chain. Then second step is somatic recombinations of a VDJ joining, variable region joining together to the diversity and joining segments. After VDJ joining, transcriptions occurs, which means DNA is copied to the primary mRNA, premature RNA. So primary transcript of the RNA is formed. Now the RNA splicing occurs, removal of the introns and VDJ concentrations get joined. So the diagram shows the VDJ concentrations. Then variable chain consisting of variable diversity joining in constant consisting of 4 constant chains. Thank you.